Good morning, all of you. I am Dr. Anand, Assistant Professor in Zoology, Devudi College, Devudi. Today we have discussing topic structure of DNA. Here, first of all, definition of DNA. DNA is nothing but the genetic information is stored, and also it is important part to the genetic material useful for the all organism. Another definition of DNA. DNA is micromolecule and they form 5' to 3' phosphodiester bond can be linked to the one another of nucleotide. DNA is made up of thousands of nucleotides. First, all DNA is present in the nucleus in all eukaryotic cells. Here, structure of DNA, the long form of DNA is deoxyribose nucleic acid. Means these are the OH group can be replaced by the hydrogen atom, can be seen to the pentose sugar. Here, components of DNA structure or composition of DNA structure includes following points. First of all, nitrogen base. Nitrogen base means these are the 4 nitrogen bases are seen to the DNA structure: adenine, guanine, cytosine and thymine. Means it is important parts to the nitrogen bases to give genetic code or genetic information of the cell. Here adenine and guanine, these are the purines. Means these are the double ring structure. These are the double ring structure, adenine and guanine. And cytosine and thymine, these are the single ring structure. Here, nitrogen bases are present to the each strand. DNA is the double helical structure. One strand is 5' to 3', and another strand is the 3' to 5'. These are the anti-parallel with each other and then connect to the phosphodiester bond. Here, purines in one strand, adenine. These are the purines, means double ring structure, and then thymine. These are the single ring structure. One strand is 5' 3', and another strand is the 3' to 5'. Means adenine double bond thymine. One strand is adenine, another strand is the thymine. Means purine binds to pyrimide. These are the double bond. Here adenine binds to the double bond. Means these are the in between adenine and thymine is considered as the hydrogen bond. Means adenine binds to the thymine to the hydrogen bond. Hydrogen bond is the weak bond. These are the during the denaturation process. Hydrogen bond is the weak and then break to separate the DNA strand. Here, next guanine and cytosine. Guanine, these are the purine binds to the cytosine. These are the single ring structure binds to the hydrogen bond, but these are the triple bond. Means these are the hydrogen bonds are connect or binds to the nitrogen bases. These are the weakest bond and they break. The two strands are separate. Renaturation, denaturation can be takes place to the during protein synthesis. Here, phosphoric acid means these are the PO. Phosphoric acid. These are the attachment to the two nucleotide Nucleotides means these are the nucleotides means nitrogen base plus phosphoric acid plus pentose sugar. These are the nucleotides. DNA is made up of nucleotide means nitrogen base means adenine, guanine, cytosine, and phosphorus group means these are the addition or connect to the pentose sugar, an attachment to the phosphoric acid, and then pentose sugar means these are the benzene ring 5 carbon atom can be seen to the pentose sugar. These are the ribose, deoxyribose sugar can be seen to the DNA structure. Means OH group can be replaced by the second carbon atom. These are replaced by the hydrogen atom. Means nitrogen base plus phosphoric acid and pentose sugar. These are made up of nucleotide. And another is nucleoside. Nucleoside means nitrogen base plus pentose sugar. Phosphoric acid cannot be present to the nucleoside. These are the nitrogen base and pentose sugar is given to the nucleoside. Here the diameter of the DNA structure, these are the 20 angstrom, and then two nucleotides. Here one nucleotide, here another nucleotide. Sugar plus nitrogen base, phosphoric acid, phosphorus group. These are the nucleotides. Here another nucleotide. These are the purine binds to pyrimidine. Two nucleotides can be bind to the phosphodiester bond. Phosphodiester bond means these are the connect one another, and long chain can be seen to the nucleotide. So these are the phosphodiester bond can be connect one nucleotide to the another nucleotide. But in DNA structure there are thousands of nucleotides are seen to the DNA, and these are the double helix structure. Means nitrogen base, phosphoric acid, and pentose sugar. These are the made up of to the DNA. DNA is the genetic material, and then transfer to the genetic information of the cell or eukaryotic cell as well as prokaryotic cell. Here pentose sugar, these are the two type of sugar can be seen to the DNA as well as RNA. Pentose sugar is deoxyribose and ribose. These are the two sugars are seen to the DNA as well as RNA structure. Deoxyribose is seen to the DNA, and ribose sugar, these are seen to the RNA structure. These are the functions of the DNA structure can be seen to the genetic information carried out, and the storage of genetic information, transmit the RNA. These are the function of DNA. The double helix structure can be discovered by the scientists Watson and Crick. These are the scientists. 1853 that can be discovered by the DNA structure. Double helix structure. These are the micromolecule, and then made up of nucleotide, nucleoside, and then all these nitrogen bases are important part to the DNA structure. Here adenine binds to the thymine, means these are the Carboprol. Carboprol means these are the amount of adenine plus amount of thymine. Means these are the in one strand 100 adenine are seen and another strand 100 thymines are seen. These are the 200. Adenine plus thymine is equal to cytosine plus guanine, or also A upon T is equal to G upon C. Means adenine plus guanine or thymine, these are the equal parts to the DNA structure. Means purine is equal to pyrimidine. Means A plus G, purine is equal to pyrimidine, means these are the A plus G is equal to 1. These are the 100 purines, 100 pyrimidines. These are the equal parts, equal components are seen to the each strand. Here in 3' end means these are the free end or these are the present OH group, and another 5' end means these are the attachment to the phosphorus group. OH group are free, these are the addition of the new nucleotide in the 3' end, not 5' end. 5' end not free, but 3' end is free. These are the addition of nucleotide. Means nucleotide chains are connected to the phosphodiester bond. Here, these are the nucleotide. These are the connect to the phosphodiester bond. And these are long chain of DNA structure. Another pentose sugar. Pentose sugar is nothing but the benzene ring. The 5 carbon atom can be seen to the deoxyribose sugar, and these are the OH group can be replaced by the hydrogen atom, and then sugar can be seen to the all DNA structure, and these are the helpful for the genetic material can be transferred. Here each term of nitrogen bases, these are the minor and major group, these are the 34 angstrom to the DNA structure. The type of DNA structure can be different, A, B, C, D, these are the type, depends upon anti-parallel, parallel structure as well as dimetron, 2-strand, another minor and major group, and then phosphodiester bond. So these are the structure of DNA is the simple double helical structure, and then micromolecule can be seen to the DNA structure, and structure is double helical, means these are the attachment of the nucleotide can be phosphodiester bond, and then 5' and 3'. 3' means these are the addition of the nucleotide. 5' these are the not free, but phosphoric acid can be attachment to the 5' end, and then adenine and guanine, cytosine. These are the number of nucleotides, nitrogen bases are seen to the each strand. These are the during denaturation, means these are the break the DNA molecule and then separate the strand, means these are the one strand is the pyrimidine, another strand is the purine. these are the separation, and then denaturation as well as rerespiration that can be seen to the DNA structure. And then function of DNA means these are the genetic material can be transferred and storage of genetic material and then transfer to the RNA of the genetic material. Here structure of DNA is the important part to the all student, and then easily describe or easily understand to the structure of DNA. It depends upon composition of DNA, nitrogen bases, phosphoric acid and pentose sugar. These are the Watson and Crick model, and the two strands are antiparallel. It is made up of two strands and attachment to the strand nitrogen bases as well as phosphodiester bond. So these are the important part to the DNA structure, and then to provide or to easily understand or objective of the DNA structure, to understand the genetic material, to understand the DNA structure and function of DNA. These are the important part to the all DNA structure of the topic. Thank you very much. I think